this is my Chapter 8 reassessment on metabolism and enzymes. So, enzymes overall make chemical reactions happen much more frequently. So, basically, we know from this that all known enzymes are catalysts, but that doesn't necessarily mean that all catalysts are enzymes. So, to start off, let's dive a little bit deeper into ATP. So, during the light reactions of photosynthesis, energy is provided by a molecule called ATP, which is the primary energy currency of all cells. Just as the dollar is used as currency to buy goods, cells use molecules of ATP as energy currency to perform immediate work because it has an unstable structure compared to ADP, making it have high phosphoryl transfer potential, meaning its tendency to release phosphate to become ADP is very high. So this is super important for many reasons. So some differences between enzymes and catalysts is that enzymes are organic, have faster reaction rates, they increase the rate of chemical reactions, and they convert the substrates into a product, and they also include activation and inhibitory enzymes. But catalysts are inorganic, have slower reaction rates, they may increase or decrease the chemical reaction rates, and they include both negative and positive catalysts. So there are 6 types of catalysts that require enzymes to move the reaction forward, and these are oxidation reduction, group transfer reactions, hydrolysis, formation or removal of a double bond with group transfer, isomerization of functional groups, and single bond formation by eliminating elements of water. Switching gears, let's talk about thermodynamics law 1. So this law is that energy can be transferred and transformed, but it cannot be created or destroyed. So an example of this to better understand is Peter eats a soft serve vanilla ice cream cone with rainbow sprinkles on it. His body transfers the chemical energy from eating this sweet treat and breaks it down into ATP, which drives his kinetic energy to ride his bike with his friend Sally later. So this example is super important, and we can see how the energy gets transferred, but it's not new energy that's creating his drive to ride his bike with Sally. However, some limitations to this include that the first law doesn't say if this process will occur on its own or not. Is it spontaneous or is it non-spontaneous? There's also no restriction on the direction of the flow of heat, so it doesn't specify the conditions that allow conversion of heat into work to be possible or the direction in which the heat transfer takes place. Thermodynamics law 2 is that every energy transfer or transformation increases the entropy of the universe. So to dive into this, an example that I thought of was Bill Nye the Science Guy put an ice cube in a cup of water at room temperature. The water released off heat and the ice cube melted in the water. The ice cube then absorbed the same amount of heat which was released by water, and so its entropy increased, resulting in a fully melted ice cube. There are also some limitations to this, is what we would think. However, these aren't really limitations, but there's a misconception that the second law is only applied to the closed system, but that's a misconception, so it's not true. However, Bill Nye came in clutch because these limitations that were in the first law of thermodynamics actually get overcome in the second law. We now know that the direction of the flow of heat helps us to predict whether the reaction is feasible or not, and it tells us that the energy cannot be completely converted into the equivalent work. Now, let's move on and talk about types of energy in the Gibbs free energy system. So, just to clear things up, free energy is not a gas station giving away free gas. However, wouldn't that be pretty amazing? Free energy process gets measured in the Gibbs free energy system. This change in Gibbs free energy during a reaction provides useful information about the reaction's energetics and spontaneity. So from this, we know that there's a bunch of different types of energy. So there could be potential energy, which is like fuel, food, batteries. There could be kinetic energy like meteors, walking, flying airplane. Cells and organisms require energy to survive, but they cannot spontaneously create it, which sort of goes back into our first law of thermodynamics and how you can't create energy. It's just a transfer. So To move on from this, cells and organisms require it, but they cannot make it. So basically this means that the chemical energy in molecules such as glucose is potential energy because when bonds break in chemical reactions, free energy gets released. And free energy is the measure of energy that is available to do work. So this amount changes during energy transfers from chemical to kinetic or chemical to electrical. So this change in energy is referred to as delta G or Gibbs free energy. Moving on, let's cover some vocab. So to start off, we have exergonic reactions. So this means energy gets released and is spontaneous, and it overall creates less product. But the opposite of that is endergonic reactions, which is when energy is needed and is non-spontaneous, so it creates more products. So in a spontaneously changed environment, the free energy of the system decreases and the system becomes more stable. And then the released free energy is harnessed to do work. So it's all about this comparison between free energy and work. That's what's highlighted here. So an example of this is water flows downhill spontaneously from higher levels to lower levels in a waterfall, but it never flows up. So this is the spontaneous process, right? Because water cannot flow up a waterfall. That just doesn't work. The whole point is it flows down. It's a waterfall. So this is a good time to mention energy coupling as well. Energy coupling occurs when the energy produced by one reaction or system is used to drive another reaction or system. To connect this to exergonic and endergonic, cells couple the exergonic reaction of ATP hydrolysis with this endergonic reactions of cellular processes. So basically, in comparing energy coupling to this waterfall phenomenon, we can tell that basically this whole definition is just that these processes go hand in hand. And I think that's really important to note in this these vocab terms end up coming up a lot throughout this whole unit, and so it's really important to understand how they go hand in hand and how they work in a system through energy coupling. So as shown above, this is the free energy equation. Basically, this tells us the enthalpy and entropy, and it tells us if a reaction is exothermic or endothermic and how organized the products are. So some more vocab got introduced, which I'll explain in the next few slides, but from this equation, we can tell that there's a lot of symbols going on. We don't really know, so I made a little bit of a key. So we know that the H stands for the enthalpy change, and that's the heat. The S stands for the entropy change, and that's the randomness, because we're all about spontaneousness, and that's really, the randomness also has to do with just the entropy in a whole system, so that's really important to note here. The G stands for the free energy change, which again goes hand in hand with the energy available to do the work, and then the T stands for the temperature. So by doing this, we can appoint each symbol to a real life thing, and it really helps us figure out the amount of free energy in a system, which is super cool because you can go in and calculate this. Moving on, it's important to note the differences in some of our vocab so we can better understand how to use it properly. So, catabolic is breaking the molecules down, like when we break down the glucose and protein in a bowl of protein oatmeal after a gym workout, while anabolic is building molecules up, like creating molecules the body can use to build its muscle mass to see strength progress. So we see how, again, this is a hand-in-hand process, much like the exergonic and the endergonic reactions, how those are an energy coupling system. It's the same situation here. Catabolic breaks down the molecules in metabolic pathways, and then the anabolic system builds these molecules back up. So again, some more vocab. We have exergonic is the net release of free energy into the environment, while endergonic is the net absorption of free energy from the environment. So ATP powers cellular work by coupling exergonic reactions to endergonic reactions. We can see how these two vocab terms, exergonic versus endergonic, go hand in hand. Again, as I was saying above, exergonic is the net release of free energy into the environment, whereas endergonic is the net absorption of free energy. So an example of this is firefly bioluminescence results from endergonic luminescence by luciferin, and this gets coupled with exergonic ATP release. So we really need both parts in the system for it to work. We can also clearly differentiate exothermic from endothermic because exothermic is the net release, while endothermic is the net absorption of heat energy from the environment. So this phenomenon clearly has to do with the temperature in some sort of way. So an example of this that I was thinking of is exothermic it could look like burning a candle because the heat from that flame gets released into the environment and warms its surroundings slightly and it melts the wax of the candle. So we can see how this entropy transfer is working through the melting candle wax. Meanwhile, an endothermic reaction could be melting an ice cube because it absorbs heat from its surrounding environment, which causes the ice to melt. So another example, if you want to see that more clearly, is a glass of lemonade with ice cubes in it on a warm summer day. So basically, endergonic would be the sun, the surrounding warmth in the environment on a summer day. The environment absorbs that into the ice, and so that's what causes the ice cubes to melt because it's absorbing that heat in this reaction. So both of these examples weren't a clear result, so we can really see what's going on here. Moving on, staying on the same topic of entropy, entropy is a measure of randomness and disorder in a system. So it's basically how crazy can you get. So an example of this, to better understand it, is there's more increasing entropy in a liquid than a solid. We know this because in a solid, everything is over-orderly and tight in a system, whereas in a liquid, the particles are free, moving, and disorderly. So I think back to middle school, biology, science, all those sorts of things when we would compare how tightly packed solid particles were compared to liquid. And even so to a gas, you see that liquid and gas, they have much more room to move around, to move in different directions, to move freely, whereas solid, you're pretty much just intact like that. And so it tells us that there's more increasing entropy in a liquid when they have this randomness and this disorder. So that gives them that freedom, and this randomness is what drives the entropy. So the entropy changes also occur in chemical reactions. So in an exergonic chemical reaction, energy gets released and entropy increases because the final products have less energy inside of them, holding their chemical bonds together. So this means that energy has been lost to the environment, usually in the form of heat. So this means that low entropy would suggest greater available energy. Meanwhile, high entropy would just indicate less energy that's useful for work in a system. Again, bringing up entropy, work, and how those go hand in hand. Just to make sure that we understand this all, a system moving towards a lower, more stable state will release heat, which increases the entropy of its surroundings. But order can't happen unless there are forces that are holding these particles together, like in the solid. So that explains why solids have less entropy than liquids. So from this, we know that water has a higher entropy than ice, so that entropy will prefer melting. Just to reiterate, energy coupling, this is the process that uses the exergonic process to drive an endergonic process, show how these systems work together, paired like that. So this happens when ATP faces hydrolysis of phosphate. This is an exergonic process, and during this time, the exergonic release of energy is greater inside of the cell due to the difference in concentration because ATP and ADP plus PI. So that, again, the concentration is super important because it shows this comparison and it shows how strong diffusion can be. And so just to kind of bring up an example to help others understand it better, ATP is like a charged battery, while ADP is like a dead battery. So ATP can get hydrolyzed to ADP plus PI when water gets added, and then it releases the energy, which turns it into an endergonic process from an exergonic process, and then ADP can be recharged to form ATP due to the addition of energy. So this is pretty cool because it's like you have ATP ready to go, but then you can get ADP to that level because of this extra phosphate in water. So this is super cool, and it shows us how ATP can be a constant energy source that works. So moving on, regenerating ATP using mitochondria in eukaryotic cells. So hydrolysis is this process of breaking complex macromolecules apart. So during this process, water gets split or sliced, as I like to remember it, hydrolysis slice, and a hydroxyl group gets added to a larger molecule. So the hydrolysis of ATP produces ADP with a phosphate ion and the release of free energy. So later on, this water gets regenerated when a third phosphate is added to the ADP molecule, reforming it back to ATP. The energy comes from the metabolism of glucose, and it shows us how ATP is a direct link between the limited set of exergonic pathways of glucose, catabolism, and endergonic pathways that power living cells. And then I have a diagram shown. Again, we're bringing up that charged battery, dead battery analogy, and we can see how above we have the ATP, and it releases energy, and it becomes that ADP plus the PI, and then it loses more water when it requires energy, and it's just this constant cycle to regenerate ATP, and it's super important. So let's move gears and talk about free energy. This is super important and was a definite highlight in this unit, chapter 8. So to start off, chemical reactions, they just need a certain amount of energy to begin working. Activation energy is the minimum energy required to cause a reaction to occur. So for this reaction to occur, existing bonds have to break and new ones have to form. A reaction is only going to proceed if the products are more stable than the reactants. Let's just let that sink in for a second. So during chemical reactions, certain chemical bonds get broken while new ones are formed. For example, when a glucose molecule is broken down, bonds between the carbon atoms of the molecule get broken. And since these are energy-storing bonds, they release energy when broken. However, to get them into the state that allows these bonds to break, the molecule must be somewhat contorted. So this means a small energy input is required to achieve this contorted state, and this contorted state is called the transition state with our vocab that we know in AP Bio, and this state is a high energy unstable state. So lots of disorder, again here, lots of entropy. For this reason, reactant molecules don't last long in this transition state, but they very quickly proceed to the next steps of the chemical reaction. So delta G, just to get in our vocab, is the change in free energy as the system goes from its initial state, like a reactant, to its final state, like a product. This delta G value tells us the maximum usable energy released or absorbed during this transition period time. And then the graph I have listed here shows us the difference in potential energy between a catalyzed and uncatalyzed reaction, and it allows us to see the timing as the free energy moves across the graph. So we start in the earlier catalyzed reaction. So you can take a moment to look at that. We have the purple line to show the catalyzed reaction and the green line to show the uncatalyzed reaction. And yeah, here we can just see how complex this system is, getting some vocab, and it shows that this cycle of reactants starts a lot earlier with the catalyzed reaction than in the uncatalyzed reaction. To dig deeper into enzymes, as shown in this picture, this is the 4-step process showing the parts of an enzyme. So as we can see here, the substrate enters the active site of the enzyme. As the substrate binds, the enzyme changes its shape slightly, and this forms the enzyme substrate complex. So then this binding of the substrate and the enzyme places stress on the bond and basically just causes the bond to break. So the products then get released and the enzyme now becomes free from its active site, so then it can go on binding to other substrates as it pleases. Now to hit lots of vocab so we can just really understand what's happening here. So to start, coenzymes are inorganic molecules that bind to the inactive sites of certain enzymes to assist in the catalysis of a reaction. They can act as the intermediate carriers of electrons during these reactions, and they can transfer between enzymes as functional groups too. Moving on, we have an enzyme. This is a protein that acts as a biological catalyst. And then moving on, we have a catalyst, which is a substance that speeds up the rate of a chemical reaction. And then we have a substrate, which is the reactant of an enzyme catalyst reaction. And then we have an enzyme substrate complex, which is a complex composed of a substrate bound to the active site of an enzyme. And then we have products, which are a chemical substance formed as a result of a chemical reaction. And then we have a competitive inhibitor, which is a substance that reduces the activity of an enzyme by entering the active site in place of this substrate, whose structure mimics. And then finally, we have the non-competitive inhibitor, which is a substance that reduces the activity of an enzyme by binding to a location remote from the active site, changing its conformation so that it no longer binds to the substrate. So part 2, just to hit some more vocab, we have the active site, which is the part of an enzyme or antibody where the chemical reaction occurs. Then we have the allosteric site, which is a specific receptor site on some part of an enzyme molecule remote from the active site. Then we have the allosteric inhibitors, which is they modify the active site of the enzyme so that the substrate binding is reduced or prevented. And then we also have allosteric activators, which they modify the active site of the enzyme so that the affinity for the substrate increases. So another cool thing that we can do with enzymes that we learned pretty well from our catalase lab actually is we can predict the activity of enzymes. So much like we made a hypothesis in that lab, here we can look at different factors such as its acidity, temperature, salt, and enzyme inhibitor presence, and we can start to make assumptions of what we think it's going to happen to it. So to dive into acid versus base, we know that we can probably assume that too much is bad and too little is bad because it always is. You eat too much food, you get fat. You don't eat enough, you get too skinny. It's always about finding that balance in life, and that is the same situation here with enzymes. So in the topic of acid versus base, an enzyme loses its ability to function when it loses its shape. So because ionic and hydrogen bonds are important factors in enzymes retaining their specific shapes, it's pH levels that interrupt this bonding negatively impact enzyme activities. Extreme pH levels may also impact the substrate or its bond, causing an even further decrease in activity. Moving on to temperature, too high of temperatures can denature enzymes by destroying both their bond and shape. So we got to see this in our catalase lab, and we got to infer what we thought was going to happen. And we saw that some of the structures and the levels of protein folding and structures were denatured because of the heat. But too low of temperatures can cause enzymes to slow down and decrease their interaction with substrates, meaning that this catalase won't really allow the enzyme and the reaction to occur. In terms of salt, if the salt concentration is close to 0, the charged amino acid side chains of the enzyme molecule will attract to each other. But on the other hand, if the salt concentration is too high, normal interaction between charged groups will get blocked, causing new interactions to occur and for the enzyme to precipitate, basically to get rid of all of that salt. It reminds me of our Patrick the Sea Star in another unit, and how that Patrick the Sea Star just had to get rid of all of that extra salt. And then finally, our enzyme inhibitor presence, they either slow down the process or stop it altogether. Inhibitors can change an enzyme shape so that it can no longer fit, which basically just demonstrates a process called non-competitive inhibition. So this is some more vocab, super important, and it helps us to make assumptions and really just see what's going to happen so we can control our results. So after this, we'll move on to feedback inhibition. So when does this process occur? So I did some research and I learned that feedback inhibition occurs when the end product of a reaction interferes with the enzyme that helped produce it. So this means that the inhibitor does this when it binds to a second active binding site that's different from the one attached to the initial reactant. So the enzyme then changes its shape and can't catalyze the reaction anymore. Feedback inhibition is used on a regular basis in cellular respiration. So this is a great example where we can see this feedback inhibition, and cellular respiration is a unit that we became very familiar on alongside Chapter 8 and Chapter 10. So this is super important. This feedback inhibition works by deactivating an enzyme using the product of the reaction the enzyme catalyzes. So basically, the enzyme binds to molecules with an active site that are specifically designed to fit with the molecule undergoing this reaction. These enzymes have a second active site for the reaction product to bind to. So this causes the enzyme to spatially rearrange so that it can no longer bind to the initial reagent and the reaction stops. So an example of this is when enzymes such as pyruvate kinase will halt the reaction. So that's just an example that happens sometimes. We've talked about pyruvate kinase, so I thought that was a good example to use, and it just shows that enzymes are pretty dang smart. And finally, another slide that we know and love, the citric acid cycle. So in this slide, you can see this picture I put here, but we're really trying to focus on citrate in the citric acid cycle. So to show how the citric molecule in the Krebs cycle illustrates how negative feedback works with enzymes, I'm going to be walking you through the 7-step cycle very briefly, as this is a pretty long presentation already. But starting with step 1 combines a 2-carbon acetyl with a 4-carbon oxaloacetate molecule to form citrate. This reaction gets controlled by negative feedback in the amount of ATP available. So the citrate loses water and then it gains another as it gets converted to this isocitrate, which is its isomer in step 2. And this molecule then gets oxidized, which is a process we're very familiar with from cellular respiration and our photosynthesis unit. And so when this happens, our NAD plus gets reduced to NADH. And then in step 3 and 4, the enzyme that catalyzes step 4 gets regulated by this feedback inhibition that we talked about above. And this is the feedback inhibition of ATP. And then succinyl in the citric acid cycle and NADH. Then after that, step 5 produces more ATP and then finally GTP. And then step 6 allows its carrier to remain attached to this enzyme and transfer electrons to the electron transport chain directly. And then this is allowed by the catalyzing step inside of the inner membrane in the mitochondrion. And then finally, in step 7, water gets added to this, and then the malate gets produced. And then the last step regenerates our oxaloacetate by oxidizing the malate, and then another NADH molecule gets produced. So this is my reassessment. I hope you enjoyed. I hope you learned a bunch of things. I tried to include as much vocab as I could. I went through all the study guides. I went through all the objectives, and I really believe that I put in as much work as I possibly could to making this is a good presentation to teach to other students. So I'm pretty proud of this. Sorry for butchering so many words. Biology words are pretty complex, let me just tell you. But yeah, I hope you enjoyed.